You're at the cutting edge of cardiovascular sciences. I'm John Cooke, professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist. And with me in the studio are two excellent guests who are going to talk about mechanisms of aging and cardiovascular disease. And to my left is Anahita Mojiri, who's going to be talking to us, who's going to be introducing our guests. Thank you, Anna.
It's my great pleasure to introduce our guests today, and actually I'm very excited to learn about their work. Joining us today is Dr. Mikhail Kolonin, professor and the director of the Center for Metabolic and Degenerative Diseases at the University of Texas, Houston. He obtained his PhD from Wayne State University, and during his postdoctoral work at MD Anderson Cancer Center, screened combinatorial libraries to identify tissue-specific cell surface markers, which have since been pursued as a drug targets for obesity and cancer. His lab investigates the roles of stromal cells in healthy metabolism and pathology, focusing on intracellular interactions mediated by GLP receptor agonist cytokines and long-chain fatty acid. His recent research highlights the roles of cellular senescence in aging-associated disease, and the title of his talk will be Tissue-Specific Functions Observed in Preventing Cardiovascular Diseases.
Pleasure to be here.
Thank you. And also joining us, Dr. Abhishek Jain in the studio. Dr. Jain is an associate professor of biomedical engineering who holds the Barbara and Ralph Cox 53 faculty fellow position at Texas A&M University. He is a founder of several next generation organ-on-chips and companion technologies of complex transport systems, AI-based analytics. He is a leader in engineering patient-specific chip organ platform for rare and orphan diseases with a focus on cardiovascular and cancer medicine. His pioneering work integrates blood and microcirculation systems in engineering cell models, advancing drug discovery, and understanding of aging, atherosclerosis, and vein thrombosis. The title of his talk is Human Vessel Chips May Serve as a Patient-Specific Models for Vascular Diseases. Thank you both for enriching our program by presenting your work.
Thank you.
Thanks, Anna, and thank you both for joining us, AJ and Mikhail, to talk about a topic that's close to our hearts, mechanisms of aging and cardiovascular disease. And each of you have done some great work in that area, and appreciate you being here today. Well, we're going to have a discussion about, first, the hallmarks of aging. So maybe we can go to that first slide and go talk through the hallmarks of aging. And what you see here is the major processes that are involved in aging. And they include, starting up there at 1 p.m., DNA damage, DNA alterations, telomere erosion, epigenetic alterations that regulate gene expression, and alterations in splicing, and proteostasis. Proteostasis is the ability of the cell to get rid of unfolded proteins, misfolded proteins, and that becomes impaired over time and can lead to protein aggregates such as seen in Alzheimer's disease, for example. So we can go around the cycle there and talk about some of these alterations that occur. Some of the other ones, mitochondrial dysfunction and generation of reactive oxygen species, impaired insulin signaling, cellular senescence, the loss of stem cells, stem cell exhaustion, and the generation of inflammatory cytokines. Dysbiosis is an interesting one that people are talking about, the aging of our microbiome, which participates in aging as well. The other way you can look at it is, different cellular compartments where things are going wrong. The secretome, what the cell makes, the inflammatory cytokines. alterations at the nuclear compartment, telomere erosion we mentioned, and disorder of the DNA, damage to the DNA, mitochondrial generation of reactive oxygen species, for example, and then alterations in the lysosomes that impair proteostasis, the ability to get rid of misfolded proteins. And let me go to the next slide there. One of the models that we study in our lab, and Dr. Mojiri has done a lot of work in this area, is Progeria. And in this slide, you see the children with the classic manifestations of progeria, basically accelerated aging, and these children look to be 80 years old when they're 8, and they die from heart attack and stroke at the age of 13, 14, 15. And the lamin mutation it causes an impairment in that protein, which is an important part of the material that holds the nucleus together, lamin A, and that protein now mutated because is called progerin. It leads to alterations in the nuclear envelope. You can see a normal nuclear envelope there in green, a nice oval, and down below it the folded nuclear envelope of a patient with progeria. And that leads to all kinds of alterations in gene expression that lead to the release of inflammatory cytokines and telomere erosion and the manifestations of accelerated aging. And one of the things that Anna has shown in the laboratory is the mRNA telomerase is capable of extending the telomere in the cells from these children. Telomere erosion, as I mentioned, is one of the major risk factors for aging, one of the major processes involved. And extension of the telomere can normalize replicative capacity in these impaired cells. In right lower quadrant there you see the impaired proliferation in red of these cells by comparison to the normal proliferation in black. And with the telomerase, you get telomere extension and you get an improvement in replicative capacity and generally functions of the endothelial cells of these children. We studied the endothelial cells of these children because they die from vascular disease, they die from heart attack and stroke. And what we find is that in addition to impaired replicative capacity, there's impaired function. They're not making nitric oxide, they're more adhesive, they're making inflammatory cytokines, and they're basically contributing to the cause of death in these kids at a young age, the vascular disease that occurs in these kids, heart attack and stroke. So let's talk a little bit about that. Let's talk about this model of aging and how it is important for us in understanding aging in general.
Yeah, I've been fascinated with this model and that work that you've done over the years, and I think it's particularly striking that by reintroducing telomerase into those cells, you can reverse those manifestations of aging.
Mm-hmm.
It
It was a surprise that because the mRNA itself is fairly transient, it only lasts for a few hours. The protein that's generated lasts for 48 hours. That seems to be enough to get some telomere extension. I think you did actually 2 transfections, so you exposed them, the cells, to mRNA twice, and you got an appreciable increase in the telomere extension. And what else did you see?
And that was enough to reversing many of hallmarks of aging, such as reducing DNA damage, such as reducing the number of beta-galactosidase positive cells.
Beta-galactosidase is what?
Which is the senescent associated
It's a marker
markers.
of senescence.
Yes, and well studied and recognized as a marker for senescence. So that is correct. The extension, the presence of the protein is short in the cells, but the effect stays long enough that is able to extend the lifespan of the cells in the cell culture, but in the mice model also extend the lifespan in the Progeria mice model. They live very short, but our therapy increased their lifespan. And when we looked at all different organs and looking at specifically on endothelial cells, we observed reversing of senescent hallmarks, as I mentioned. And that was interesting because of 2 reasons. First, all of us, while this is a very specific aging disease associated with a mutation, but all of us by aging, because of missplicing, we do have expression of progerin. So it is a good model to study. And the other reason is this reversing stayed long. And that was also, and without actually fixing any mutation. The telomerase doesn't fix the mutation, but it seems that reduce progerin and that fixing the nuclear morphology, which associated with other recovery that we have seen in the cells and in the mice.
Thanks. AJ, you have, you'll be talking a little bit later about your model using a kind of a cell-based chip model. What have you done with respect to aging in that model?
Yeah, I've been fascinated by, Progeria cells as a model to look into the aging mechanisms, and then, one of the opportunities we have now relative to 10, 20 years ago is that we have much more sophisticated in vitro model systems that can be much more physiologically relevant. And I'm particularly impressed to see how this field has grown and converged by including not just cells but also the physical factors, as well as the biochemical and molecular factors that can be dissected and analyzed using these modern tools. So, in my lab we have been trying to use this as a bottoms-up approach to see aging mechanisms and to test these novel therapies. I've been particularly impressed how this mRNA therapy since the COVID times has advanced. Circular RNA, which is now under development, is also a very promising area, and I think you're working in that too. So we think that these model systems now and in the future will really bring the therapies to the clinic much faster than they used to.
Mikhail, you use animal models for your studies of aging. I think you have a few slides to show. Would you like to talk about that? Maybe we can go through Dr. Kolonin's slides.
Absolutely, yeah. So what my lab's been working on is the mouse model of aging, and as Dr. Cooke and Dr. Mojiri nicely introduced, cell senescence is one of the hallmarks of aging. So as we grow older, we accumulate senescent cells, and the definition of senescence is an ability to continue dividing. And when stem cells become senescent, of course, the whole organ falls apart. And senescence arises for many different reasons, but telomere shortening, DNA damage in the nucleus, and DNA damage in the mitochondria, all of those things are important. And endothelial cells are the cells that are integral to our health, and those are the cells in our blood vessels which have the first impact, because all of the xenogens that we intake with our diet, with the toxins from the environment, endothelial cells are the first one to see them all, and they get damaged the most. And high glucose, which arises as a result of metabolic disease that becomes more apparent as we age, all of those cells take it on, and eventually, as you can see on the slide, if you take out endothelial cells from an old person, they just look different. They look large, the nucleus looks large, and they become more fibroblastic. They don't make contact with other endothelial cells as well, and more of those cells accumulate in our blood vessels. What we don't know right now is whether this process is different in different organs. You could imagine that some of our organs take on more impact than others, and we decided to make a mouse model to study that. But before I get to this, so what was really telling in your presentation is that when you reintroduced telomerase into the cells, you reversed several different hallmarks of aging. And I think it's very important because aging biologists keep on debating, which hallmarks of aging is the most important one. Is there a trigger that makes everything fall apart? And the fact that you can reintroduce telomerase and reverse a number of hallmarks of aging tells us that perhaps telomerase is quite pivotal in those processes. And again, to reiterate the point that Dr. Cooke already made, telomerase extends the pieces of DNA on the ends of our chromosomes so that cells can continue dividing. Without telomerase, telomeres are not extended, and at some point after every division they become shorter and shorter. So stem cells at some point will sense that the telomeres are too short to divide. It's dangerous because there will be chromosomal fusion, and they'll stop, and this is a terminal event. You get senescence. So, and the cell has a choice of either die or just sit there. And unfortunately, we accumulate those cells that just won't go away. They just sit and they secrete those toxic inflammatory cytokines and molecules that harm other cells. What's been discovered is that this enzyme, telomerase, abbreviated as TERT, it does other things that are very important in addition to extending telomeres. So it turns out that TERT works as a transcriptional co-regulator throughout the genome. It binds to transcription factor machinery, and it modulates how transcription factors such as TGF-beta, Wnt, beta-catenin, MYC, those important factors that are important for cell proliferation, how those are operating. And also, TERT turns out to be getting out of the nucleus. It goes to the mitochondria, and it plays an important role in the mitochondria, and it protects mitochondrial DNA, apparently through mechanisms which we don't completely understand. And most of this work has been through cell culture. And when we work with cell culture, as scientists, we know that a lot of results that we get, could be artifacts. So it was very important for us to use animal models to find out which of those tentative mechanisms are true. And we decided to test this hypothesis that telomerase in vivo in animals is not only important for telomere extension, but also potentially for other non-canonical function, without which endothelial cells don't function well and age faster. And we made mice in which telomerase was knocked out in endothelial cells specifically through this Krylok system, and it's an endothelial cell specific promoter that we used to excise telomerase specifically in cells, in endothelial cells of the mouse. And at the same time, the cells in which this happened, the endothelial cells, they switch color from red fluorescence, they switch to green fluorescence, so we can track those cells as knockout cells. And what we found out at the bottom is that indeed green cells, when you take them out of the mouse, they don't proliferate well, they change their phenotype, so clearly there is senescence. And as you see in the middle at the bottom, when you put cells into those 3D vascularization assays, you can see that they're incapable of forming normal vascular structures. And also through in vitro and in vivo experiments, we found that blood vessels in those mice with knockout of telomerase in endothelial cells, they become leaky. So if you inject a dye, it will penetrate into the parenchyma much faster in knockout mice. And this is in part because endothelial cells become become hypoxic. And this is in the graph in the middle on the right. So we measured HIF-1 alpha, a factor which is a marker of hypoxia in endothelial cells expressed in GFP, and we found that it's markedly upregulated. So there is this dysfunction of vasculature in those mice which manifests in multi-organ dysfunction. So when we looked at the function of various organs of those mice, including the brain, the skeletal muscle, the adipose tissue, we saw manifestations, we saw the phenotypes which are characteristic of aging, and this is just one phenotype. So there are two mice on the left that can run really well when they're electrocuted a little bit, they're stimulated to run forward, and the two mice on the right, which are the knockout, even though they're electrocuted, they become fatigued and they get exhausted much quicker, and they fall behind and they cannot run anymore, which tells you that telomerase is important for the function of skeletal muscle cells, and you wouldn't be surprised to find out that it's also important for the function of smooth muscle cells in our hearts.
That's really interesting. On that last one, you're showing that the animals in which you knocked out TERT, their endurance is reduced. Their ability to run on a treadmill is reduced. And you mentioned that, maybe because there's some abnormalities of the skeletal muscle, which is probably true. But also, have you measured perfusion in the skeletal muscle? Are those mice getting less perfusion because the vasculature is aged?
So this is really interesting, Dr. Cooke. So we did measure perfusion in those mice, and to our surprise, perfusion is actually fine. And what the interesting phenomenon that happens in
Now, let me ask you a question. Was that perfusion measured at baseline or with exercise?
It was been measured at baseline. We did not
Because
measure.
it could be that the exercise-induced increase in blood flow, which is necessary for them to be able to run, is impaired. We did show that limb blood flow with exercise is highly dependent upon endothelial release of nitric oxide. That's impaired with aging. And so it could be that the exercise-induced increment in blood flow is impaired.
That's a great point. And so what's interesting is that we looked at their lymphatic circulation, and if we inject a dye subcutaneously at the bottom of the foot, or then we see that at baseline lymphatic circulation is really inefficient. But the blood circulation at the baseline is fine. But you're very right. I think it's the challenge which they're not doing well with. Our explanation is that it's the mitochondrial function in endothelial cells and in myocytes as well, which is affected. And what's really interesting in those mice is that even though the knockout of telomerase is specifically in endothelial cells, when we looked at other cells in every tissue, in the skeletal muscle and adipose tissue, we found that other cells in the tissue, they become affected and they undergo so-called juxtacrine senescence. They induce markers of senescence and they become also metabolically dysfunctional. So I think it's a combination of processes that happen in those animals that probably simulate what happens in us as we age and as our endothelial cells become senescent and all cells in our organs become dysfunctional as a result.
That's a really great point, because if your endothelial cells are senescent, then they're releasing inflammatory cytokines that make the cells around them sick.
Yeah, just emphasizing on that point that this is very fascinating work, and that shows how important to take care of our vasculature, our endothelial cells. So meaning that all you need to get old is to have old vasculature, and if you get to that point, perhaps by high blood pressure, high cholesterol, high glucose, that may happen easily for the vasculature, and that could be the initiation for the other cells to become senescent.
And the other way around, hopefully, if you restore the function of endothelial cells, if you reverse their senescence, if this is possible at all through approaches such as those that you're developing, perhaps we could fix all the other cells in our organs as well, or at least in our hearts.
Let's hope so. AJ, do you have a comment on that?
Yeah, no, I find vasculature very fascinating too. We talk about, organ systems and how cells assemble to become tissues and organs. But vasculature is an organ, but it's not an independent organ in the sense that it intersects with every organ of our body. So it's part and integrated to every organ of the body. So, as Anna just mentioned, you fix the vasculature, you fix the disease, but not only you fix the disease, you improve overall health and not just part of the health. So I think this is very important research, and these are very important outcomes that we have achieved, and there's a long way to go in terms of finding treatments, but I think fixing the vasculature for aging is something that's going to drive the next phase of medicine.
What do we know about that, AJ, about the different processes of aging and their importance for different individuals? We did see that telomerase and the telomere seem to be very important.
Yeah.
It's a major determinant of aging. But is it possible that some people age differently?
Yes, absolutely. I think sex and race and other demographic differences could influence how you age, and the mechanisms that regulate aging might also be different amongst patients, not just at an individual level, but also among different ethnic groups, etc. So I think it would be very interesting to see how different groups age differently, and predisposed conditions, for example, diabetic patients or cancer patients, how does aging intersect diabetes and vice versa, in the same way as other diseases like cancer and vice versa. So I think aging in its intersection with different sets of people, different sets of other diseases is also something that we need to look at.
Maybe this would be go ahead, Mikhail.
Yeah, there is just one comment that I want to make. So telomerase, since we started talking about it, is an interesting protein which is different in mice and in people. And in us, in humans, telomerase is turned off. In embryonic stem cells it's on, but then in somatic stem cells, in adults, telomerase is turned off. This is the dogma. In mice, it's expressed through life, which is why the knockout that we made shows the phenotype. Otherwise, it wouldn't have shown the phenotype. So that's the distinction. But there is more and more evidence accumulating that in people, telomerase could be turning on in certain conditions, and that could explain, or could be expressed at higher baseline levels in some individuals compared to others, and that could explain as to why different humans age differently, and could potentially explain why exercise is such a powerful anti-aging approach.
I'm glad you mentioned that, because we have data suggesting that telomerase is reactivated in somatic cells with injury. So if you injure the skin of a mouse or a pig or a human, we now have data, right? That telomerase is reactivated at the edges of the wound and may be involved in the regenerative response.
And there are papers that show that it's turned on by exercise as well, and time will tell whether those papers are for real, but we're working on it too.
Yeah.
Interesting.
Since we're talking about exercise, I've been very fascinated to look at cardiovascular disease and aging amongst astronauts who go to space and come back with cardiovascular disease. And I think it would be very interesting to see how we can apply these models and these approaches into treating and preventing astronauts who go to space and come back. Given that space travel is going to become privatized, we are planning to send private citizens to space. So we want them to remain healthy when they come back. So I think there's enough opportunity there to look into the health of those people who are going to space and coming back.
The astronauts need an exercise pill.
Yeah.
We all need an exercise pill.
Yeah, absolutely.
I actually have a quick question. That could be a question for our audience as well. You mentioned that human, as they grow, they stop expressing telomerase, but in mice, they keep expressing telomerase. So how come then the mice live just 2 years, considering with us that we live relatively very
Great
long?
question. So obviously telomerase is not the only explanation, for better or worse longevity. There are other things at play. Mice, to put things simply, they have a lousy, defense against damage. They're missing certain enzymes which humans express at very high levels, so it's a trade-off. Mice have longer telomeres, they express telomerase, but cell repair machinery is much weaker, and that's a short answer to your question.
And gets back to this idea that different organisms, but different individuals, humans, may age differently. And one person that could help us get, I don't think we're going to talk about this particular aspect, but you could look at this with your model system. Maybe we should talk about your model system now.
Yeah, absolutely.
And because you have the ability to look at different individuals or different conditions and their effects on aging in human cells with your chip system.
Absolutely, yeah. So, my lab works at this intersection of tissue engineering and microfluidic science. And we work with what we call as vascularized tissue systems, or what we simply call them as vessel chips. The idea is that you can grow vessels or more complex vasculature in these very small microfluidic cassettes that we call them as organ chips. And then you can use that to culture, living human cells, and then you can look at a variety of questions in different contexts, including aging and atherosclerosis. So, when we think of engineering, aging or atherosclerosis with organ chips, one of the values that I see that these kinds of vessel chips that they can bring to the table is that it's a bottoms-up approach. Unlike our animal systems, which is a top-down approach. You take a complex system in an animal and you try to reduce it to a certain level of simplicity to ask specific questions. These in vitro systems are a bottoms-up approach where you build complexity to a level where you can ask that same specific question or a different question. So I think these model systems can collaborate with each other to really, influence, what we can learn from not just at a disease level, but also at a patient-specific level, because you can get, very specific patient cells. Progeria is an example. You can use progeria cells to get to a level, at a resolution where you can identify mechanisms that are regulating a specific patient using these model systems. So I believe that, these organ chip systems have value. The other value is the nexus of different variables that control this complex disease. When you age, your vessels become stiff. When your vessels become stiff, the flow is no longer the same as it was for a healthy patient. Okay, the vessels themselves are not just straight tubes. They are branches. They can converge, they can diverge. All these different shapes and architecture of the vessels, they can twist and turn. And they can become more twisty vessels as you age and as they become stiff. How does that flow influence or regulate some of these mechanisms that we want to understand? And how can these treatments ultimately can influence and restore not just one cell, but the entire function or the circulation or microcirculation of a patient, is something that you can start thinking of modeling with these systems. So in my lab, we create these systems that we call as these vessel chips. These are different types of designs. You can apply them for different questions. Some are single vessels, some are more complex vessels, some are capillary tubes, etc. And these endothelialized vessel chips have another advantage, that they are very simple to make, because you can create a hollow tube. You can culture endothelial cells in 3 dimensions. You can put extracellular matrix around it, and then you can put flow or even blood cells through that. So now you have this model where you can very systematically look at endothelial cells, its interaction as with the matrix, its interaction with the blood cells or immune cells, as you say, and learn quite a bit of it. And you can make these chips as healthy controls, or you can take patient cells to make them models of aging or atherosclerosis. This is just another video of what it looks like from the inside. This is a 3-dimensional model. What you see here in green are all endothelial cells. They can form a cylindrical tube, but for simplicity, you can also create elliptical tubes. Not all vessels are cylindrical. They can be elliptical as well. So you can create these elliptical vessels. What you don't see is the matrix around it. We can also co-culture them with perivascular cells or, muscle cells or pericytes to look at, what happens in aging as endothelial cells age. How do they interact with the perivascular? Or when the perivascular cells age, how do they interact with the endothelial cells? Underflow. So again, these are some examples of what you can achieve. I have a portfolio that we have built in my lab where you can not just make straight vessels, you can actually make these living endothelialized structures that are far more complex, like, for example, branches, or tortuous vessels, or vessels that have the shape of stenosis. You can co-culture them with muscle cells. One of the unique things that we have learned is obviously that hemodynamic waveforms that can be very complex. They could be biphasic, triphasic. They have a time lag. So hemodynamics itself is so complex, and each part of the hemodynamic system, the amplitude, the frequency, the time lag, that could also be influencing how the cells behave. So we have this toolbox now where you can actually dissect the physical forces, the biochemical components of a tissue or a vascular tissue in an organ, to look at very specifically how these mechanisms are influencing aging, and then how they can be reversed with therapies like the mRNA therapy. Another advantage is obviously the analytical tools that we have now. We can intersect these tools with omics analysis. So, for example, you could do very high quality transcriptomics, proteomics, even metabolomics, to actually learn about mechanisms from these human cells in an organ level context. We have also worked with now machine learning and artificial intelligence to automate it. And when you automate it, you can hand these tools to users who are not experts in these omics. You can get the data that you want much more quickly, and you can make complex decisions very, very quickly using these model systems. So these are some examples, where we have used patient-specific cells in creating these patient chips. iPSC-derived cells is a wonderful opportunity to actually get patient-specific cells. But there are other areas in the body where you can actually mine these cells. For example, we've harnessed blood-circulating endothelial cells, also called as also a subset of blood outgrowth endothelial cells. And what we have learned is that you can take blood samples from humans, about 30-40 ml of blood. You can take endothelial cells and you can culture them and then make vessel chips out of these cells. You can take the blood from that same patient and circulate the blood in the same patient chip, and you can actually start learning about the disease. So for example, we have what we call it as a diabetes chip, where we took these cells from a diabetic patient and created a vessel chip of a diabetic patient and started looking into very specific vasculopathic outcomes of a diabetic patient, and the diversity that you were just mentioning between, for example, males versus females. We can start to learn from it. So as a proof of concept, we have shown that you can create these, what we call as a diabetic patient chip. We've also worked with sickle cell disease chips. These kids who experience pain, the only treatment that they have is a hydroxyurea treatment. We really do not know which of these kids, for example, have a propensity to get a stroke. And a large number of kids land up getting a stroke when they are sickle cell disease patients, but not all of them. So is there a way where these tools can actually serve as a predictive model, where we can learn a little bit and start learning whether this particular kid can have a stroke or not? To be able to do that, we need to look into the vessel, but we cannot do that in vivo, right? And it's very hard to also create an animal model of a sickle cell patient with pain. These are very difficult models to create. So we took, these blood cells from sickle cell patients. We harnessed the endothelial cells and we made these sickle cell patient chips, and we actually started seeing, the vasculopathic, responses in these vessel chips amongst a subset of these patients. Are these subset of patients have a propensity of a downstream stroke? We don't know that yet. We haven't gone that far yet. But we now have a system where you can potentially ask these questions. So I'm pretty excited by the value that these kind of technologies bring to the table.
That's actually really fascinating. I mean, when we think of sickle cell disease, we think of the hemoglobinopathy that leads to sickling of the red blood cells, and in the microcirculation leads to occlusion of the microvasculature. But what you're saying is that some patients may be resistant to that or more susceptible to that based upon their vasculature, based upon their endothelial cells, because at that level the microvasculature is basically endothelial cells forming those capillaries.
Yeah.
So what you're saying is that if you have endothelial cells that are resistant, that child, that patient with sickle cells, much less likely to have a problem. If we understood the mechanisms of that resistance, maybe we have another therapy for sickle cell. That's really interesting.
Yes, absolutely. For the longest time, sickle cell disease has been seen as a blood disease. Even if you want to learn about the sickle cell disease in medical schools, you have to go to the department where you will have to learn about bloods. So it's really cardiovascular medicine or cardiovascular scientists don't really talk much about sickle cell disease. But what we're now learning is sickle cell disease is also a vascular disease, right? And the endothelium is very, very involved in it. So it's very interesting, how different parts of our body, they interact with each other. The blood cells interact with the vasculature and vice versa. So I think, in aging as well, and when we talk about aging, when you talked about different tissue systems, the vasculature and its intersection with the liver, vasculature and its intersection with the pancreas, with other organs, with the lungs, is also very important. And to start looking at the vascular disease in that particular context is, I think, very important.
Fascinating.
Yes, I think it's a fascinating technology, and I think what would be just looking into the future, I think the challenges that we're facing in biomedicine is identifying the patients who have endothelial dysfunction, and again, endothelial dysfunction may be organ-specific, tissue-specific. You may have perfectly good vasculature in your skeletal muscle, but your vasculature in the heart may be affected for whatever reason, or so organ specificity is the key. And induced pluripotent stem cells are a fantastic, tool to, for the proof of principle, but it doesn't tell you about, the site specificity of the patient, what is happening to the vasculature, in this particular place at this particular moment. So probes to identify dysfunctional vasculature, identify those senescent cells, is one thing that we need to develop.
Oh, that's interesting. Yeah,
Yeah.
that's an interesting idea, right? Because what you're saying is that in the same individual, organs may be aging at different rates.
Yes.
And you can think of the person who's a smoker, right?
Yeah.
And you can actually see that they're aging a little faster around their eyes and their
And
mouth.
then the lungs, obviously.
Obviously their lungs.
Yeah.
Here's another thing, that if you're talking about blood vessel aging, blood vessels age faster at bends, branches, and bifurcations, right? Where you see endothelial cell turnover is occurring faster. And those are the sites where atherosclerosis occurs, vascular aging. I want to turn back to Mikhail for a moment to talk about his. I just want to make sure you were done though with your slides.
Yes, absolutely. I was just about to end by saying, we have reached this time where we can start looking into, vascularized systems and starts looking at aging and disease as a systems disease.
Yeah.
So your point is spot on, and I think we need to look at it from that perspective.
Thank you. That was great. But back to bends, branches, and bifurcations for a moment, where vessels age faster. You see, that's where the lesions of atherosclerosis occur, at
That's
those
right.
sites. And you have some insight into that from your work with macrophage. Do you want to talk about that? We've been talking about endothelial cells, but we
Yes.
should talk about another
Yes,
cell
yes.
type.
Yes, I'll at least mention that. But before we get there, I want to kind of reiterate another point. So the one challenge is to identify dysfunctional vasculature, where and when it comes about, and the second thing is to do something about it. What do we do? What do we fix in endothelial cells? And from your fascinating work, reintroducing telomerase is one thing that we could potentially do. And from our studies, I don't know if the slide deck is coming on again, we've been looking at what breaks in endothelial cells without telomerase. And what I was going to show in my presentation is that in those mice where we knock out telomerase in endothelial cells, when we look at skeletal muscle, the telomeres are perfectly fine. They're not shorter, which is something that you could potentially expect, because endothelial cells don't divide all that much, right? So it's the cell divisions that shorten the telomeres, but endothelial cells, they're formed during development, and unless it's tissue such as adipose tissue where there's a lot of remodeling and growth where telomeres can erode, because stem cells have to make more and more cells. In the skeletal muscle, it doesn't really happen, and in the heart it doesn't really happen. So when we looked at the muscle tissue, telomeres are fine, yet cells are dysfunctional. And I think we're coming to a slide where we found out that the senescence of those cells happens because, here you go. So what happens is that the graph shows that telomeres are fine, but nonetheless cells are metabolically defective, and specifically their oxidative metabolism is hammered. So the seahorse analysis shows that they consume less oxygen, and I cannot advance for some reason. Maybe you can advance for me. But what we found is that when we looked at gene expression, we found that markers of oxidative phosphorylation were generally down, and as a compensation, markers of glycolysis went up. So endothelial cells switch their metabolic program to survive. There you go. So this is what's shown there. And also, we measured in the middle on the left the numbers of mitochondria that the endothelial cells have in those mice with endothelial knockout of telomerase. And what we found that is that mitochondrial numbers are lower. So this oxidative defect in those cells of those mice is because there are fewer mitochondria. And basically this is the conclusion of this aging cell paper that we published last year, which is that telomerase, the non-canonical function of telomerase is to protect mitochondria, probably by protecting their DNA. We don't know exactly what's going on, but without telomerase, mitochondria undergo damage, they die, and without that, the reactive oxygen species and oxidative damage becomes unleashed, and that hammers the nuclear DNA, and the problem happens because of that.
So let's talk about that for a minute. Anna, looks like you have a question.
I actually do have a question. So when we talk about telomerase, it's well accepted and commonly known that it extends the telomeres to protect the telomeres by extension, right? Now, there are published data, including yours, that there are localization of the telomerase in the mitochondria. And now that the defect that you observe here, do you think it's start with the shortening of telomere and then affect mitochondria, or it initiate in the lack of telomerase in the mitochondria that affect gene expression in the nuclei? Which would start?
So from our data, the indication is it's the latter. So without any problems in telomeres, we don't know. There could be telomere damage. This is the thing. So all we know is that telomeres are not shorter in endothelial cells of those knockout mice. Telomeres, though, they are the sites where DNA damage accumulates more. So telomerase, the enzyme, indirectly through gene expression regulation, it supports gene repair machinery, and without telomerase, one thing that happens is that there is more DNA damage without telomerase. So without telomerase, mitochondria are dysfunctional and are going away because there is damage there. Nuclear DNA is damaged more, and telomeres may look fine, but
it's a vicious cycle, isn't it? Because if your mitochondria are dysfunctional, they're generating more reactive oxygen species. That leads to more DNA damage, nuclear DNA damage, as well as mitochondrial DNA damage and accelerated aging.
It is a vicious cycle, indeed.
Yeah.
So one thing that I wanted to finish with, if I have a minute or two, is this model that we haven't published yet. So along the lines of the work that you've reported, we reintroduced telomerase through a transgene into brown adipocytes, in this case, in cultured cells. And what we discovered is that some of the oxidative phosphorylation genes have been induced by telomerase, and also mitochondrial numbers were induced, as you can see on top right. And when we subjected cells to ethidium bromide damage, ethidium bromide is an agent which damages DNA. What we found out is that ectopic expression of telomerase suppresses the DNA damage which is incurred by the cell. So it's really important, and it's really promising, your work and our work, that it could be a protective approach.
Do we know how telomerase gets to the mitochondrial DNA? Are there some accessory proteins that guide it to the mitochondrial DNA?
So this is the black box. No one knows. It's not clear which genes it binds to, if any, whether it's at the level of there are some papers that indicate that it binds to mRNA, and that's how it affects mitochondrial health. But it's all speculations. We don't have any solid proof.
And I guess we don't really understand the mechanisms by which it's restoring mitochondrial function.
Correct.
Mitochondrial DNA. Do you actually have evidence that mitochondrial DNA damage is repaired by the
So we don't have
telomerase?
this. No. So we don't have this direct evidence. But what we do see is that cells are more resistant to ethidium bromide-induced damage. You have more mitochondria basically surviving. So we don't know if it induces regeneration of mitochondria, but at least we have more mitochondria, so it probably prevents their disappearance. That's our hunch.
Interesting. And you did say that the telomerase can have an effect on nuclear expression of mitochondrial genes that are important for.
So we didn't specifically look at mitochondrial genes, and you've done a much better job. In fact, the genomic analysis that you did. So I think all of us need to look at. Well, so I take it back. We did look at the expression of mitochondrial genes, and yes, indeed, when we knock out telomerase, we do see the reduction in the expression of mitochondrial proteins expressed by the nucleus. That is correct.
It's actually very interesting. I don't know if Dr. Jain, can you detect mitochondria also in your vessel chip? Is that it's more being used as the effect on cells function, but I'm not sure if I have seen any publication looking at the mitochondria in the vessel chip.
I think it should be doable. I mean, it's the biomarkers that you would use to detect mitochondria would still remain the same. You just need to put it in the microscope and see the process. So
it would be actually very interesting using the endothelial cells lack of telomerase putting in the chip, because yes, your work nicely showed that when you don't have a telomerase, you are accumulating all aging associated markers in the cells. But now the question is whether all of them will reverse if you add telomerase. Is all of them reversible, or some of them are is at the end of the road, they cannot reverse? So that would be interesting to look at endothelial cells.
That's right. So I think we are going to learn a lot from gene therapy approaches, and endothelial cells. The beauty of them is that they're accessible to what you put into circulation, and they will receive gene therapy, telomerase gene therapy that you're working on. The other cells that are accessible to gene therapy vectors that are introduced are leukocytes, such as macrophage, and as Dr. Cooke mentioned, our unpublished work that I can present real quick.
Yeah, let's take a look at that.
Indicates that senescence that occurs in macrophages, in myeloid cells, monocytes, that make foam cells in cardiovascular disease and atherosclerosis are so those changes arise because of telomerase inactivation as well. So what we haven't published yet is a model of mouse monocyte-specific telomerase knockout, and this panel shows that we see those beta-galactosidase positive cells, senescent cells. They we just lost something there. So just in the nutshell, what we see again, just like in endothelial cells, oxidative metabolism in monocytes and macrophages goes down, glycolysis goes up, and the very important phenotype that we saw in those mice is heart disease. They develop right-sided ventricular dysfunction. Their cardiac output is much lower than in wild-type mice. We did it in collaboration with Jagat Narula and Eva Sevic at the University of Texas Health Science Center. And the underlying cause of this is twofold. First of all, those macrophages without telomerase that undergo senescence, they become more pro-inflammatory polarized. Macrophages can be either classically polarized to pro-inflammatory or alternatively. So these are classically polarized a lot more, and they accumulate a lot more lipid droplets. So they actually form, they turn into foam cells systemically. And those are the cells that make atherosclerotic plaques. And the other thing that happens in those mice is that they have a lot of fibrosis in both in the heart and in the lungs. And the combination of those fibrotic conditions can manifest in the heart disease, because there is a lot of pushback from dysfunctional lungs into the heart on the right side, and this is probably what develops in this really clinically relevant heart phenotype.
Yeah. Wow, this is really a great insight, because as you pointed out, foam cells are the elements that compose the fatty streak and ultimately grow into an atherosclerotic plaque. Their accumulation in the blood vessel results in atherosclerosis, foam cells. And what you see in the diagrams when we see someone talking about atherosclerosis, right, you see monocytes adhering to the endothelium, going into the vessel wall, absorbing lipid that has found its way, because a person is hypercholesterolemic, found its way into the vessel wall, it absorbs that, they become foam cells. But they never leave. It's like Hotel California. You can check in, but you can't leave. They just stay there, and they accumulate. And I'd always thought, and people had said, that the reason they do that is because, well, the person is continuing to be hypercholesterolemic, cholesterol's coming in, and they basically, it's a process that's self-propagating. But what you're saying is they become senescent. And as they become senescent, they start to behave like senescent cells and make a lot of inflammatory cytokines. And what's that's going to do? That's going to call in more leukocytes.
That's right. And the question, in this study and in all of the studies that people do on senescence, what's the chicken, what's the egg? Do they become senescent first and then they become dysfunctional, or they become dysfunctional and then they become senescent? Or those two things happen, in parallel that we don't know.
Yeah.
One thing we didn't talk about is therapeutics, and you just reminded me of the work that James Kirkland is doing at Mayo Clinic with senolytic strategies. So using drugs to actually kill senescent cells and get rid of them. That could be potentially very useful in a lot of diseases, but particularly atherosclerosis, I think.
It could be. The devil's advocate argument is that you get rid of those senescent cells, but they're senescent for a reason, and they have to be replaced with something. So if you have, telomerase inactivated, if telomeres are short, then there will be a void which will not be filled with anything good potentially. So that's the catch.
Filled with more senescent cells. Maybe.
Exactly. That's the catch.
Well, we're coming to the end of the program, but I'd like to give everyone a chance to make some final comments. So, AJ, are you ready to make a few comments about aging and the vasculature, and what you think are, what you plan to do to provide more insights and potential therapeutic avenues?
Absolutely. I think, my opinion, I think this convergence of different experimental model systems, animal models, and as well as these bioengineering approaches is a very promising direction. We know that FDA Modernization Act that was passed by the Congress, the 2.0, there is a 3.0 in the works that is going to be allowing, these non-animal approaches to work, in tandem with other model systems to speed up potentially bringing new therapeutics for complex diseases like aging and atherosclerosis. So I think the time is right. I think for looking at aging and atherosclerosis from different perspectives that we talked about, the diversity of the disease, both inside the body, amongst humans, etc., and bring different approaches to solve this complex problem.
Mikhail, I'm going to let you have the last word.
Thank you. Well, so I have this telomerase-centric view on everything, and because telomerase is so potent in suppressing or even reversing senescence, I believe that this is the future, and we need to keep on synergizing in our work on figuring out how to reintroduce telomerase and how into our cells, into our vascular cells, into our leukocytes, and how to rejuvenate our cells through those approaches. And there are some small molecules that have been reported to activate telomerase. Obviously, gene therapy is another approach, and there are pros and cons to each, but at some point there should be clinical trials done that will test those the viability of them.
That's exciting. Well, thank you, Professor Kolonin and Professor Jain, for joining us on the show today. And thank you, Dr. Mojiri, for being my co-host. And thank you all for joining us today at the cutting edge of cardiovascular sciences, where we talked about aging and cardiovascular disease.